we have digested, and I hope, don't forget here, in the test, in your lecture test on the digestive system, I remember asking about substrate, the enzyme, and then the products, right? So you have your, you remember we had your polysaccharides. Don't forget this term. What is this? What does it mean? Polysaccharides is your?
Multiple sugars.
Yes, your carbohydrates, right? And then your polypeptides. This is your?
Proteins.
Yes, and then your lipids, right? So they entered my GI tract, and through combination, mechanical and chemical digestion, I now have glucose, right? I now have amino acids, and I now have my free fatty acids. Don't forget, in the test, we talk about protein digestion, and remember, we talked about the proteases, which are the protein digesting enzymes, and you have two locations for proteases. Where do you find proteases? In the stomach, it's called pepsin, right? And in the small intestine are the other proteases. The only similarity between the two of them is their secretion in zymogen form. Remember that? They're in active form. But in the stomach, how do you activate them? You need acid. In the small intestine, how are the zymogens activated? By the enterokinase, remember, an enzyme. So the only thing similar is their secretion in inactive form. Where's the site for amylase in carb digestion in the body? Mouth and small intestine. Where's the site for fat digestion? In the small intestine. So here, you digested it in the mouth and small intestine. Here, in the stomach and small intestine. And here is just only the small intestine. Okay? So this now, and then these cells, they eventually ended in our cells, right? So once we absorb them first, they enter in the submucosa. You have your blood vessels and your lymphatics. This glucose and amino acids will enter immediately my blood. They end up in the plasma of my blood. And then they get transported, then they go to the cell. They go inside the cell, right? And then my free fatty acids, first, they enter my lacteals, which is your lymphatic capillary in the GI tract. And then this is where they get combined to a lipoprotein form, a water-soluble form, because fat is insoluble in water, remember? And that's the main component of our plasma. If this join the plasma right away, it will separate, and it's not good for the blood. So it has to first enter your lacteals, and it must be in the lacteal that it will be converted to a water-soluble form, so then it can join your blood and that way enter your cell. So this chapter talks about when this chapter talks about when these nutrients.
What happens to the polypeptides?
Huh?
You have the glucose going into your blood, and then you have your free fatty acids.
It's also into plasma because there's a little line.
Oh, the plasma, okay.
Okay. So we're going to discuss what happens when these cells enter the cell. No, not cells. When these nutrients enter the cell. So we started with our 4 groups of food for you guys. Your hamburger. These are the grease, salt, sugar. This is just something fine. Anyway, so this
Chocolate's not on there.
Oh yeah, I should have.
Sugar was on there.
So we eat, we digest, we absorb. Then what happens to the nutrients? So they are called nutrients. So these nutrients that we're going to study are the energy yielding nutrients, meaning your carbohydrates, your proteins, and your fats. They are referred to as energy yielding nutrients. There are other nutrients that don't give you energy in the form of calories, like your vitamins, right? Your water. They're also nutrients, but you don't get calories from them. This chapter talks about how do we get energy from these nutrients, okay? So this is a basic overview of how we get energy from nutrients. all books always talk about carbohydrate digestion first. And there must be a reason, okay? I'm just going to draw my cell again. You follow? You get to the part to why they entered my cell. Remember for glucose, remember they entered the cell, but remember that you have insulin is the key. Remember insulin is the key to let them in. And then the other nutrients, they just follow. They just ride on the uh, glucose car, let's say. They just ride, and that's how they enter the cell. So glucose, amino acids, and my free fatty acids eventually end up inside the cell. And I want to know, how do I use them? What is their purpose? Why do we need them? The main purpose for glucose is to give us energy. Remember when we were looking at chapter 3 by 241, we said, what's the function of glucose? For energy. No, that's chapter 2. It's for energy. It's your main source of energy. And then we said, what's the function of fat? It's your stored energy. Okay? I remember in your test, I talked about this is stored energy. And I have these three. You don't forget that you also have stored glucose called glycogen. So you will choose this and this as stored. So the difference between the two, they're both stored, but this one is to be used also now. So there's two forms of glucose or carbs inside your cells. The one that you're using now and the one that you're storing. You store it in your liver and in your muscle, right? And then the amino acids, you're not meant to use it, right? It's meant to, once it enters, remember? So once you have all these amino acids in the cell, immediately they're going to be used, right? For protein synthesis. You're going to use it maybe to repair your cell membrane, maybe to make enzymes, to make a hormone, whatever, to make your oil glands, to make your mucus, to make collagen, whatever. That's the purpose of amino acids. But I want to look at the metabolism step by step. Why do we use glucose always? Why is it the first one that we burn for energy? Oh, and also in this story of nutrition metabolism, we're going to include one very important organelle, which is our mitochondria. Okay?
ATP.
That's correct. So ATP. ATP is the only energy currency of the cell. So all cells, to do their job, let's say this is a cell that has to make enzymes, or it has to make collagen, or whatever it needs to do, it can't do it unless it has ATP. Or maybe it needs to make neurotransmitters. It needs energy to do all those things. So the energy is coming from first glucose. So the question then is, why is it always glucose that we always burn? Even though you have glucose, you have fatty acids, you have your amino acids in here. They're all, oh, by the way, one more thing. Don't forget the terms. This is the cell membrane, right? This is the cytoplasm and the nucleus, okay? So they all are present in the cytoplasm. Why do we always burn glucose?
It's the easiest.
The what?
It's the easiest.
It's the easiest because of enzymes. Very good. If I had enzymes to burn these fats and the amino acids, then I can burn them all at the same time. But in this, remember, okay, you remember that the mitochondria is for ATP, right? But there must be something going on in the cytoplasm that makes my cells burn the glucose first. Do you remember the term glycolysis? Where we break the glucose in half. And you break that, you release energy, a little bit of energy already. That event only happens in your red blood cells, with glucose, because of the fact that there is an enzyme. See? There's an enzyme that will break down the glucose into two. It's called glycolysis. So this one right here, releasing pyruvic acid, and that enzyme is called hexokinase. Do you understand? You just want to, so immediately, in other words, what I want you to know, so they end up here, glucose, amino acids, and free fatty acids. For these two, the amino acids and free fatty acids, so that I can use it for energy, this has to be transported here. You have to wait for it to get to the mitochondria. Whereas the glucose, it already is met by enzymes in the cytoplasm once it enters. So you can burn it for energy. Does it make sense, everybody? Okay? So that's why, that is that explains why glucose is always the first nutrient that we burn through glycolysis. And so that's why all books on metabolism always start with glucose metabolism. Okay? If I ask you in the test, why does metabolism always begin with glycolysis? Even though all three of them, all three nutrients end up, they're all present inside the cell. You're going to talk about two things. One, mitochondria, and the other one, cytoplasm. So when they all enter, they are met with enzymes already. But these enzymes only target glucose. So you're already getting energy from glucose through glycolysis. For these two, there's no enzyme that meets them. The enzyme to break them down is here in the mitochondria. So they still have to go there. Okay? Okay? So the first step is called glycolysis. By the way, before we go over this one, how did we form our glucose? How did we form our fats? Remember that this is you, right? So you breathe out carbon dioxide in the air, right? In the meantime, here's your plants, right? So the plants will use the carbon dioxide plus the energy from the sun, right? And then here you are, you're watering your plants. You water your plants. So the carbon dioxide plus the water plus the energy from the sun, energy, that's what gives you carbs, C6H12O6. When we eat this plant, right, we get the carbs again. And then we process it this way, and then we breathe it out. It's just a cycle. Okay? So, now let's look at first glucose.
Oh, darn. I was
What?
trying to copy that.
The drawing?
Yeah.
The person? You breathe out carbon dioxide, and then the carbon dioxide will be used by the plants, and here's you are watering your plants, right?
Mhm.
So see CO2 and H2O with the energy from the sun will combine them. That's the energy. So the sun energy, solar energy. Solar energy, you have your C6H12O6. Oh, that's what's being stored by the plant. And then when you eat the plant, you break it down again to release your glucose. Okay? So let's look at this glucose metabolism. The first step is always glycolysis. By the way, there's something very important, okay? We know that glycogen or glucose, okay, when glucose enters the cell, this is very important for you to know. The glucose that enters the cell is locked in. So there's no way for it to escape and be free. It's just once it enters, its fate is sealed. You're going to burn it for energy. It's locked in the form of G6-phosphate. So when you immediately, it gets phosphorylated, in other words. That's what happens. When glucose goes in, it gets phosphorylated, and in this form, it can go out anymore. Because you may wonder, or I guess a lot of the physiologists or scientists before, they wonder, if glucose is able to freely enter, when it goes in, it's going to be concentrated here. How come it doesn't come back out? Does it make sense? It was first concentrated here, went down here. If it went there, then that means it becomes concentrated. How come it's not able to go back out? And then they realized that that glucose, even if it's getting to be more and more concentrated here, it gets locked in this form. You phosphorylate it, it's a bigger form, and it's not able to go out. So what happens is we burn it. So here's your glucose. This is another way to write the glucose formula, right? C6H12O6. And through glycolysis, where is this happening? You should know where this happens, in the cytoplasm. Okay? So the process of glycolysis breaks this down into two pyruvic acid molecules. So this is pyruvic acid. But when you broke the bond, you release the energy. So there's already energy. And that's, when you do your weightlifting or resistance exercise, that tides us over for about half a minute, okay? But after this one, with pyruvic acid, this is called, did you notice we didn't need the mitochondria for this one? That's why this is referred to as anaerobic metabolism. You understand? Okay? But this pyruvic acid, in the absence of oxygen, then will enter your blood and be converted to lactic acid, right? Which can cause muscle cramping. So this converts to lactic acid. And this can cause muscle cramping. This can decrease the pH. You need to remember, okay, in your test, that was just a taste test of a pH, acidosis. So pH is always going down when conditions are being more and more acidic. Don't forget that. Because in your next test, acid-base balance, even here, I think I talk about pH again. But more so in acid-base balance test with your renal system. Okay? And so this lactic acid can decrease pH all over, and acidosis is just not a good condition, right? Just think of you as meat. You're just like pigs. We're just like chicken. We're animals. And you know that to tenderize the meat of animals, or whatever meat that we're trying to tenderize, you soak it in, marinate it in vinegar, and that will tenderize the meat. And that's what's going to happen to us. So we cannot afford, and that's why we stop exercising when we go into acidosis. We get that fatigue. So to get more energy from this one, you already know that we have to send it to the mitochondria. So the mitochondria is the last stop, and this is where it gets converted to its breakdown products of carbon dioxide, water, ATP, and heat. We're going to talk about, I'm going to write it down, okay? But what is the event that's going on in the mitochondria? You get this part, don't forget to write down, this is the cytoplasm, right? And you understand why glucose is always the first one we burn. Here in the cytoplasm, they are met by enzymes that can only target your glucose, not your amino acids, not your free fatty acids. They're safe, okay? So you're immediately breaking this down, yielding pyruvic acid. And this pyruvic acid, in the anaerobic pathway, you just need lactic acid. But in the presence of oxygen, this can go into your mitochondria. Okay? What is that event going inside the mitochondria? That event is called Krebs cycle. There's one more very important term I want you to remember for this slide, okay? So see, there's a few problems, right? Here, your problem is enzyme, so that's why you can only burn glucose. Here, there's a problem. The mitochondria only accepts this acetyl-CoA. This is the only substrate that the mitochondria can use to give you ATP. Okay? Not glucose, not fatty acids, not even this pyruvic acid. So this has to be converted to acetyl-CoA. Okay? If you want to use your amino acids or your fatty acids, they have to be converted in this form, acetyl-CoA. Okay? And so that's the first step. As this pyruvic acid enters the mitochondria, it undergoes decarboxylation. I hope you understand this term, just breaking down, right? So metabolism is just a series of decarboxylation, just breaking it down, the carbon molecule at a time, decarboxylation. You understand decarboxylation? You break it down, one carbon molecule at a time. So again, what substrate does the mitochondria recognize only? Acetyl-CoA. That's the only substrate that the mitochondria can use to give you ATP. But when this pyruvic acid entered your mitochondria, it's in this form. It's in the pyruvic acid form. So the first step is to decarboxylate and convert it to acetyl-CoA. In acetyl-CoA form, it can now undergo the Krebs cycle. The Krebs cycle is the main cycle here in the mitochondria that all our cells use to get ATP. This is the most efficient way for getting the maximum amount of ATP from any nutrient. Okay, for our purposes, as far as I'm concerned, I do not require you to know all these different substrates, okay? And this is also called the citric acid cycle. Look at this part over here. Don't forget, see? This was pyruvic acid. This was what happened from glycolysis, right? We get pyruvic acid and there was ATP here already in the cytoplasm, right? And then to take advantage of this event called Krebs cycle in the mitochondria, look at the first step. We had to convert it to acetyl-CoA. So this pyruvic acid is 1, 2, 3 carbon molecules long. This one is just 2 carbon molecules long. So you decarboxylate, and now in this form it can enter my Krebs cycle, which is what is going on in the mitochondria. And so the way I try to analyze this citric acid cycle is just to think of it as like a subway, right? Where there's a series of stops in the subway, the door opens. So it is nothing but, look at, it's nothing but a series of decarboxylation reactions. So you're breaking it down, one carbon dioxide molecule at a time. You have to do it step by step. Why? Because if we broke all the bonds all at the same time, a lot of that will be wasted as heat. We want to conserve as much energy as possible and channel all of that into forming your ATP. But if we broke them down all at the same time, you're going to waste most of that. You will still get ATP, but a lot of it will be wasted as heat. There is heat already being wasted here a little bit still. Do you understand? Again, am I requiring you to know? No, it's too much for you to know. But what I want you to grasp is what is going on. Okay?
Can we go back to through glycolysis, you're breaking down glucose. So how are you burning it and bringing it down at the same time? I'm confused.
Burning is breaking down.
Okay.
When you burn something, you're breaking it down.
Right.
I'm using them
And
interchangeably.
it's being converted to pyruvic acid.
Yes,
That gets sent to the mitochondria.
And then it's sent to the mitochondria.
Okay.
Look at this. There's your
Alrighty.
glucose. There, see? This was my glucose.
Okay.
This was my glucose. And this is in the cytoplasm. This is in the cytoplasm. So you broke it down into two. This is the glycolysis part. Then you end up with pyruvic acid. So next, in the cytoplasm, we have this organelle called mitochondria. So I'm going to send this, if I want to burn this more or further, which I should because there's still energy there, you see, right? What, to combine two molecules isn't there energy, right? So I still want to take advantage of that, but I cannot do that if it stays in the cytoplasm. So the next step is to send it to the mitochondria. And what is going on in the mitochondria is this one. So what's this one? It's decarboxylating. See? In other words, breaking that bond. You break that bond to release energy. But as you break the bonds, there are the electrons that's being exposed. Okay, you remember how atoms look like, right? You have your protons, neutrons, and then you have the electrons. You remember the octet rule? Remember? And so there's electrons that's being exposed, and there's still energy there. So the last stop is to send the electrons to the electron transport chain, which is in the mitochondria also. So again, this metabolism in the mitochondria for humans, even for animals, it's a step-by-step process. The reason they do that step by step and it's not all of them at the same time is because you're going to waste a lot of it in the form of heat. In this way, you do step by step, you're able to really very efficiently have all the energy that's being released and convert it to ATP. Do you understand this slide? It's very easy, isn't it? Okay? Just don't be overwhelmed with all of those substrates right here. Right? Isocitrate, then citric acid, then don't be overwhelmed with those substrates. Just look at, like I said, just look at it like a subway, right? Where in each open exit, you see there's a passenger coming out. So that's the decarboxylation. So you started with these two carbon molecules right here, the acetyl-CoA, and then you release them. See? And then the rest of the electrons, you channel them to the next, which is called the electron. Where is all of this event? Where are all of these events happening? In the mitochondria. So what does the mitochondria need so this can happen? Oxygen. The reason why you need oxygen is because remember, that's the nature of atoms. You cannot, atoms are not meant to have electrons exposed. They have to satisfy their outermost shell, right? And so that's why you have this terminal electron acceptor. It accepts. So what will happen? These electrons, by the way, are bound to your protons, your hydrogen, right? So what will happen is hydrogen will combine with oxygen to form your water. So this water is like the end, and it has been divested of the energy. All of that energy was channeled to forming your ATP. You understand?
No.
No?
Can we have a slide for dummies, please?
Again.
Wait, so I just have a quick question. For the electron transport chain, that's before the ATP, because like I was just
Your
confused.
ATP is your last step, or the very last step?
I was just confused where that was thrown in. I was like,
No. It's a step-down process. You're slowly harvesting all the energy to form your ATP.
Okay.
Slowly. Again, here's our glucose. 1, 2, 3, 4, 5, 6. So we understand why glycolysis is the first step, right? And this is in the cytoplasm. Okay? Break it down. You produce pyruvic acid, which is how it looks like, right? So if I want to use this, I have to decarboxylate it first. You're releasing the carbon dioxide. You can send it now to the mitochondria. So now all of these events from here on is happening in the mitochondria. Okay? So in acetyl-CoA form, I can send this to the Krebs cycle, which is just an event. It's called a Krebs cycle because it was Mr. Krebs, oh no, Dr. Krebs that discovered this cycle. Like how they wanted to know, how do we get ATP? They just wanted to know. So here is a series of substrates right here. By the way, this substrate, this citric acid, this fumarate, all of this, these are by the way sugar substitute. They're carbohydrate forms. Okay, that's why you cannot burn energy in the absence of carbs. And you know this one, this NADH, this flavin, these are coenzymes. They're called coenzymes. They're needed for metabolism to happen. If you, in case you didn't know, these are your B vitamins. That's why they ask you to take your B vitamins for energy. Okay? And what do you mean by that? It means that they're necessary so you can break down your nutrients. You get your ATP. Okay?
That slide makes my head hurt.
This slide here, oh, I'm just going back. You don't need to know.
Thank you.
Okay, so you understand this part right here, right? So we jumped in the Krebs cycle transportation or subway, that's how I try to call it. And it's a series of decarboxylation. So you see, you just break down. You break down that, you break down that. But as you break it down, you're releasing the atoms with the N plus the hydrogen protons with the exposed electrons, right? So what are you going to do? There's still energy there. You send it then to the electron transport chain. These events happen in that mitochondria. It looks like this, right? Those events happen in the mitochondria. So eventually, this electron, so it's a step-down process where you harvest the energy from these electrons. But the electrons, they have to go somewhere. And so they eventually bond with oxygen, which is the terminal electron acceptor. That's how, and these are the electrons that are in your hydrogen atoms. So they bond with oxygen to produce water. So you have divested all the energy in that glucose molecule and transferred that to ATP. By the way, not all because there's heat. So we're going to look at the final breakdown products.
Okay, the glucose, it has a little arrow that points to the ATP and the...
Yeah, that's glycolysis.
Okay, that's glycolysis.
Then you have pyruvic
okay.
acid. See,
Okay.
number one, glycolysis.
And then the pyruvic acid, one goes to the Krebs cycle. What happens to the other one?
Both
There's
of
another
them.
arrow. What's
Both
the
of
other arrow then that goes to the middle?
This one here?
No, the purple arrow that points over to the right. The CO
This one here?
something.
That's the two.
That's the decarboxylation. Remember,
Oh,
the
okay.
mitochondria can only accept acetyl. That's the only form that it can process, acetyl-CoA. Don't forget
Okay.
that for the test. Right? It can only process or accept acetyl-CoA. That's the only substrate that the mitochondria can use for this.
So if that's all glucose,
Yes.
since we've sent the other ones straight to the mitochondria, do they pick up at the acetyl-CoA?
That's very good. That's what we're going to look at. How they have to be converted to us. Are you talking about the fats and the yes.
And the proteins.
Yes. We're going to talk about that next. Very good observation. So for your nutrients to be used, they have to only be in acetyl-CoA form. Let's look at the final breakdown products. Do you understand these final breakdown products? Look at the final breakdown products of energy metabolism. You have carbon dioxide. You understand this one, right? Right? You have carbon dioxide. That's a breakdown product of metabolizing your nutrient. You have water. You get that? You understand why we have this, everybody? How about why we have this? Yes? And then we also have ATP. You understand? Carbon dioxide plus water plus ATP. You need to add in your notes plus heat. There's always some of it released as heat. It is not a perfect 100% efficient. converting of all the energy to ATP. See, even though you do it step by step, you're still already losing some of it as imagine if you broke all of them, all of those bonds at the same time, you're going to waste a lot of it.
What's the NADH2?
Those are your coenzymes. This is going to be
Coenzymes.
transported here. Okay? So, next will be your fats. Do you have any questions on carb metabolism? Now let's look at our fat metabolism. Okay? Oh, this is a nice summary, by the way. I like this slide. This was lifted from Tortora's book, but you have this in your book too. But I like it because I don't know, it says cytosol, and then it has the nice picture of the mitochondria. I like pictures. And here, don't forget to circle this acetyl-CoA. It's only in acetyl-CoA form that the mitochondria can work for you. Then you have Krebs cycle, electron transport. I like this. Okay? Am I going to, I know that you're going to ask me, am I going to require you to memorize this number of ATP? No. For me.
I don't think I have that picture.
It's not. Maybe for chemistry you need to, but for
Rubic
me,
acid.
I just want you to really understand why, and the basic. Because there's too much. This is slide number 25. Okay, next.
No, I don't have that picture. Where do you have that?
It's a different. It's chemistry.
Oh,
But you have a nice picture there.
where?
In your handout or the slides.
I thought you have it
Okay.
in your, I will check. Okay. Next, we're going to look at lipid metabolism. Okay? The next is your lipid metabolism. So here, Calista, look at this one. See, look at this. To use your fats or your amino acids, you cannot, there's no separate breakdown of fats or amino acids in the cytoplasm. So I can use them, I have to send them to the Krebs cycle. And to take advantage of them, they have to be in acetyl-CoA form. That's the form that they have to be. Okay? Now, let's look at the next one that we use. It's fats. That's the one that we burn next, right? If we use up all our carbs, we now turn to our fats. The first very important thing that we need to do for that fat is to convert it into a water-soluble form. That is the only way that it can enter the cell, right? When I absorb it from my GI tract, it is in free fatty acid form, and this is insoluble in fat. So, in my lacteals, you remember your lacteals? As it enters the lacteals, it will already be converted to a lipoprotein form. And please write down in big letters in your notebooks. All lipoproteins are soluble in water. I can also say that all lipoproteins are soluble in blood. But the plain free fatty acid form, the plain cholesterol, insoluble in blood. So these are the 4 lipoproteins that we are going to talk about. This chylomicron, VLDL, LDL, and HDL. I know you know this, the HDL and LDL. This is your good, this is your bad. People always get confused. So how do you remember H for heaven? For me, that's how, and heaven is where good people go, is what they say. Okay? So that's how I try to remember. This is your good cholesterol, and L is the bad cholesterol, in case you forget, okay? Okay, so again, these are the fat forms. These are all the lipoproteins, all of them soluble in blood. Because I remember there was a test where there was a matching type, or ABCDE, remember, multiple choice. Soluble in blood. All of them are soluble in blood. So let's look at how they become this, how they became this. So first, we start with the fat that entered my lacteals. Okay?